So the work I presented at ARUK is kind of one aspect of the ongoing work in my lab, which is an interest in tau proteostasis. Obviously, we're interested in tau protein because it's a core component of the pathology of Alzheimer's disease. And what we've tried to focus on in my group, aspects of the tau protein that we can't easily model in other systems. So a good example of that is tau splicing. Tau splicing is very complicated. It's also very species specific. So the tau splicing in humans is different to what we see in rodent models. And so we've been trying to build human models that splice tau in the way that we see in the human brain. The reason that's important is because we know that tau splicing is critically linked to neuronal health. So mutations in the tau gene that disrupt tau splicing can cause frontotemporal dementia. So it's really important that we have a model to capture tau splicing and to capture the consequence of disruptions to tau splicing. And the challenge of doing this in the human model is that tau splicing is also developmentally regulated. So there's a transition from fetal stages to postnatal stages where the splicing changes. And we've been really interested in whether the stem cell derived models could recapitulate this. And, you know, the short summary of that part of my talk is yes, they can, but it takes a long time. So we're invested now in doing these long-term cultures over, you know, periods of up to 300 days where we really see a developmental transition in the way that tau is kind of produced and spliced by the cells. And this finding really prompted us to ask other questions about what is different in how the cell processes tau in early development compared into adulthood. And we're now very interested in this idea of how tau proteostasis might change. So the cells break down proteins, they turn them over kind of as needed. We're interested in this idea that perhaps these very young neurons that we're growing in the lab are really well equipped to break down abnormal proteins compared to in adulthood, when obviously we see these proteins start to accumulate. And there's a term that I really like, which I didn't come up with, it was from the literature, but this idea of super vigilant proteostasis in early development, which of course makes sense because these are very young neurons. They will live for the lifetime of the person. So if there's an aggregate or something abnormal forming during early development, that might then compromise the long-term viability. So what we found in the lab are things like proteasome activity is really, really high in early development. But also if you interfere with proteostasis using, for example, proteasome inhibitors, certainly in our model, that doesn't seem to lead to tau aggregation. It actually seems to, the cell can respond to it in a flexible way. So it can kind of sense that the proteasome isn't working and upregulate other degradation pathways to cope with that. So we have this idea that if we can understand what regulates the turnover of tau in early development, that might give us insight into strategies that could then be protective against tau aggregation.